Welcome to Chapter 1 of our HDX DIA educational series. In this session, we'll focus on the science behind hydrogen deuterium exchange, HDX, paired with mass spectrometry. You will see how this powerful technique reveals which parts of a protein are stable, which are flexible, and how proteins interact with one another or with small molecules. We will walk through the entire process, starting from labeling proteins in heavy water, then digesting them into peptides, and finally analyzing those peptides using mass spectrometry. By the end of this chapter, you will have a clear understanding of how HDX works in practice and why it is such an essential tool for investigating protein structure and dynamics. Let's get started.
Hydrogen deuterium exchange paired with mass spectrometry, or HDX-MS, is a technique that we use to study proteins. As you may know, proteins are not static systems. They have movement. Inside of a protein, some parts of the protein may be static and may be moving very little, like vibrating very little, whereas other parts of the protein might be moving quite a lot and might be involved in actual protein function. HDX-MS is able to give us an idea whether this protein, which parts of it are active and which parts of it are buried away in structure. And this can be used to study, for example, a protein system where multiple proteins are interacting with each other, as well as a protein interacting with a small molecule target. The way that HDX works is that a protein is exposed to a D2O solution, which is just heavy water where the hydrogens are replaced by deuterons, which are heavier counterparts. And the heavy water will exchange certain hydrogens on the surface of a protein to deuterons. So for example, if this is the protein sequence, the backbone hydrogens in the protein sequence can exchange for deuterons in areas that are easily accessible to the solvent and not so much in areas that are not easily accessible to the solvent. After labeling, the protein can be digested using digestion enzymes to generate peptides, which peptides will then carry the respective local deuterons that have exchanged at that particular location, and these peptides can be measured using mass spectrometry. In a mass spectrum, an unlabeled peptide, or one that does not have any deuterons, will appear at its normal location with its expected natural isotopic abundance, as shown in black. After the peptide is exposed to deuterons and it's carrying some of the deuterons from the protein sequence, that peptide population will now appear heavier on average than its non-deuterated counterpart. And that's how we can tell how many deuterons on average that peptide is holding as a population. That is measured by taking the centroid of the mass of the labeled population and subtracting the centroid of the non-labeled population. Typically, this is done as a single measurement on a mass spec where we take a peptide in a run and just measure its mass difference. After we calculate the mass shifts for every peptide in our protein system, we can do this at different multiple labeling time points just to get an idea of the exchange profile of a certain part of the protein.
In this first chapter, we explored the fundamentals of hydrogen deuterium exchange and its role in revealing the dynamic nature of proteins. In Chapter 2, we will delve into the effects of collision-based fragmentation. You will learn about hydrogen scrambling, why it occurs, and how it can be utilized to generate additional measurements from a single peptide. Let's move on.